Hello, this is your lecture for Chapter 10, the senses. We're going to be covering the basic senses as well as some special senses. So, basic introduction, senses are derived from sensory receptors. We learned about those in that last chapter. They're detecting any changes to our environment, internal or external, and they send signals to the central nervous system for interpretation. There are two types of senses, general and special. General senses touch, pressure, temperature, and pain, all distributed, very structurally, very simple, not a complex process, versus the special senses, which you might kind of think more as your senses, vision, hearing, smell, taste, and balance. No science yet on the sixth sense. So it's senses use action potentials, but we're aware of different sensory events because of different types of receptors. So the types of sensory receptors, based on what they're detecting, their sensitivities, what they're sensitive to. You have chemoreceptors, changes in chemical concentrations, things like blood glucose, carbon dioxide, oxygen, those types of things. Pain receptors are nociceptors, detecting any damage to our tissue. Thermoreceptors, responding to temperature. Mechanoreceptors, to pressure or movement. We'll see different types. We have those through our GI system and our muscles. And photoreceptors that respond to light. So the difference between the sensation and perception. So sensation is when receptors are stimulated and send impulses to the brain. And then perception is our awareness of that stimuli. I always say, I've been a massage therapist for a long time, and a lot of times I'll get different reactions, either pain or laughing, tickling reactions based on how people perceive the actual sensation that they are receiving. So the perception of a signal can be dependent on sometimes just our own personal wiring. So as a sensation is being formed, the brain uses projection to send a sensation back to its point of origin. This helps us to pinpoint where something is. So projection is allowing us to know, oh, the fly, I feel a fly biting on me. That's the perception. Oh, I know where it is on my arm. That's projection. And yes, sensation depends on the region of the brain receiving impulses. So adaptation is something that our nervous system goes through. We are not always consciously aware of every stimuli that's coming to us. So the brain prioritizes incoming sensory impulses to prevent us being overwhelmed by that incoming information. It can become less responsive as well to a maintained stimulus. A common one is your clothes. When you put those on right in the beginning of the day, or whenever you start your day, you will feel them rubbing up against your skin, but then even after just a few seconds or a minute or so, you get used to that sensation of them. This is also that thing that, we adapt to our normal, and so people will say, they have no idea that they were even in pain until they got out of pain, because that was just standard to them. This is called sensory adaptation. So this is the ability of the nervous system to become less responsive to a maintained stimulus. So it's not just that we don't perceive the stimulus as well, it's that our bodies don't send as many signals, because they start to say, okay, well, this is just normal. This is just, you get aware of it right away, but, you then adapt to it. This can either be, again, from unresponsive or inhibitory pathways. So general senses, I'm getting into those. They are widespread. They have receptors in the skin, muscles, joints, and viscera. These are your organs. Senses for touch, pressure, temperature, and pain. So touch and pressure are sensing deformation or displacement of tissue. So as you go to touch something, the shape of whatever it is that you're touching stimulates these receptors in specific ways that helps you determine the shape, the texture, the hardness of your grip, the firmness of your grip, rather. So those free nerve endings that we spoke about, tactile or Meissner's corpuscles, sorry, these are responding to motion of objects that contact the skin. A lot of times in hairless areas, think about your finger pads, lips, those types of places. And then those lamellated corpuscles, deep in connective tissue, they detect that deeper pressure in the dermis and subcutaneous. Here's just a picture of that. You should recognize this from our integumentary lectures. Temperature receptors, thermoreceptors, two groups of free nerve endings in the skin, warm and cold. So they respond based on what the temperature is, and as they do adapt quickly, but they will still give us an intense stimulus, to let you know very rapidly, oh, this temperature is changing. They give you that burning sensation or the freezing sensation, that cold burning sensation. Proprioception. Proprioception is the ability to know where you are in space. This is what gets inhibited in your medulla oblongata when you are intoxicated or under the influence. Proprioceptors, that's a tough word sometimes. So these are the receptors for proprioception. They are in skeletal muscle, in the muscle and in the tendons. So muscle spindles, these are bundles of skeletal fibers within skeletal muscle that are monitoring the state of muscle contraction. So they're determining how strong, how much effort or force is being applied. And then Golgi tendon organs are in the tendons where muscles attach to bones, and they're determining the amount of stretch that a muscle is going through. So if you try to pick up something too heavy, let's say, you for some reason need to pick up a car and you try to go pick it up and it's too heavy for you to lift on your own, the muscle spindles will basically shut off your muscles to make sure you don't overexert. And the Golgi tendon organs are for stretching. So if you ever stretch and you start to get to that point where you feel like you're about to tear it, again, your muscles will kind of automatically contract, reflexively contract, based on a stimulus sent from this receptor. Here you can see the muscle spindle fibers embedded within the muscle fiber, the muscle belly itself, versus the tendon organ, which is in the actual tendon. Nociceptors, your pain receptors, they have free nerve endings, is one type, as well as things like cold receptors, if they, or those temperature receptors, if they get overstimulated, that can be interpreted as pain as well. There's also some local signaling that each cell can do to kind of signal pain, inflammation, that stuff. So nociceptors communicate with pain sensory neurons via neurotransmitters, either substance P or glutamate, depending on the location. So those nociceptors will synapse with the spinal cord and release those different neurotransmitters to stimulate a pain response or pain perception. Tissue damage also causes what we call prostaglandins to be released. So if actual cells are damaged, then they will release this prostaglandin, this local signaling hormone, and then that will bind and stimulate nociceptors and increase that sensitivity to them. Remember, there's different ways for the nervous system to be stimulated. You can also have factors that basically just make your nervous system cells a little less negative. So they don't maybe make it reach threshold, but they get it there a little bit easier. So that way, if there's other cells that are experiencing pain and also releasing prostaglandins, it helps to make it even more likely that you'll get a nervous system stimulus. Things like aspirin, ibuprofen inhibit prostaglandin synthesis, so that's why they're, they don't actually resolve the issue. Taking aspirin and ibuprofen don't solve the problem. They actually just inhibit our sensation and awareness of it. Morphine, heroin, natural painkillers, things like endorphins, they inhibit that substance P release to help, again, dulling pain sensation, pain sensation. And since the pain is advantageous to health, it warns of possible danger to the body. The whole theory of no pain, no gain is not true. Pain is there as a way to tell you that you are about to hurt yourself, you're about to do serious damage. And so it's important to listen to that. It's important to make sure, usually most physicians will tell you that if the pain is not over-manageable, if it's not too high of a fever, if it's not too much sinus pressure, allow your body to go through natural processes, because inflammation, swelling, that type of stuff is part of the healing process. And when we immediately try to mitigate those symptoms, these secondary symptoms, pain, swelling, that loss of motion, that kind of stuff, it can inhibit the healing process. So visceral pain, these are again in the viscera, so in our organs, producing sensations. They respond differently than those on the surface. In some cases, damage doesn't produce pain sensations. You hear all the time of people having these fairly progressed injuries, internal injuries, and not knowing it. And it's because our body doesn't feel pain inside like the way that we do outside. Pain comes from mechanoreceptors, decreased blood flow, oxygen deprivation, or chemical stimulating pain receptors, chemoreceptors, sorry. Referred pain is also very common. This is when you have pain in your viscera that feels like it's coming from a different part of the body. And this is because of common nerve pathways between the skin and internal organs. One example is the heart is often felt it's coming from the left shoulder or the left arm, particularly in males, not always in females. This is just a map of this, so you can see it's very interesting. I definitely recommend taking some time to look through it. But all the different spots on the body where, if you are having pain in this organ, you're going to be feeling it. You could potentially feel it in these areas. Some are pretty broad, like the kidney, covers a large swath of tissue. But then something like the appendix is almost always going to be felt, not always going to be felt, I wouldn't say that, but it is very commonly felt in this lower right quadrant. So that's always a good spot to be aware of. If people are having consistent increasing pain in this lower right quadrant, it's good to have them checked out. Maybe take them to the doctor just to make sure. Take them to the ER if they need to be, especially if it's progressing and not moving. If the pain is staying in one area, yeah. These are also not completely diagnostic, obviously. These are just ways that you can use to help try to narrow down where things might be occurring. But they are very consistent throughout people because of those common nerve pathways. There is just an example of that if you'd like to see a pictographic representation of the sensory fibers from the heart and the skin meeting in this spinal nerve. All right, so let's get into special senses then. These are a little bit more complex because they don't just have a simple signal transduction. There's actually a way that they are themselves stimulated. And so we'll go through smell, taste, hearing, equilibrium, and sight. Starting with smell, olfaction, that is the sense of smell. So the olfactory organs are masses of epithelium in the root of the nasal cavity. They have these olfactory receptor cells, so masses of epithelium in the roof of the nasal cavity. So if you think about that, that's the very, very top of your nose, the top of the nasal cavity. The roof of it is lined with an epithelial tissue, so a covering tissue, the cellular tissue, and dangling from that are these little olfactory receptors, and they kind of hang at the, just like a little chandelier or something, just kind of hanging down right at the top of the nasal cavity. So that way they can detect scents. And that's why when you breathe in deeply, you might smell things more intensely because more scent molecules are being, those receptors in the roof of the epithelium are being stimulated more. So the olfactory receptors are bipolar neurons that are covered with cilia. They have one type of olfactory receptor membrane protein that they're releasing. Olfactory receptors are chemoreceptors. Inhaled odorants stimulate particular groups to provide a discrimination of smells. So this is not a mechanoreceptor. It's not a photoreceptor. It is a chemoreceptor. It's responding to dissolved liquids, dissolved chemicals. Senses of smell and taste function together to help with food selection. So when you can't smell, things don't taste quite as the same. So here you can see this is the cribriform plate of the ethmoid bone that sits in the skull, and these little olfactory receptors are dangling down, and they're able to synapse with chemicals, these chemoreceptors. And here you can see it from the sagittal view, where you have the nostrils, air moving up into the top of the nasal cavity, and then the brain. So olfactory stimulation, each odor stimulates a set of specific receptors. The receptor cell membranes of olfactory receptor cells causes an influx of sodium ions for depolarization, so that's pretty standard, to reach threshold, and then the action potential gets generated. Hundreds of types of olfactory receptor cells, they code for thousands of odors and transmit signals to the brain group. So depending on the specific combination of receptor cells that are stimulated by those odorant chemicals, our body, that's the sensation, and then the perception in our brain is what the smell is. That's why sometimes you can smell one thing and it's like, wait, is it that or is it this? And until you see it or someone tells you, oh, it's that, your brain doesn't fully recognize, oh, that's what it is. Okay. The brain interprets different receptor combinations as an olfactory code, and they adapt very quickly, so smells fade quickly. That's why, just going back to this quick, that's why when you smell something really quickly just without having to inhale deeply, and you still smell it for a long time, in your brain you kind of know, oh, that's a strong smell, versus something where you might have to really, really sniff deep and smell deeply, and then if you do smell it, it goes away very quickly. It's, oh, that's a very faint smell. That depends on the density of these odorant molecules. Okay, taste buds. So sense of taste. Taste buds are organs, the organs of taste, about 50 to 100 taste cells, mostly on the papilla of the tongue. So this is the little projections of the tongue, those bumps. And they're scattered throughout the mouth and pharynx. So the actual taste buds themselves, the organs, those are microscopic, right? Even though you might still feel some ridges and bumps on your tongue, that's the papilla, that's not the actual buds themselves. So taste cells, your gustatory cells, are modified epithelial cells that function as chemoreceptors and get replaced every 10 days. These are it's a very highly abrasive area on the inside of the mouth. All that chewing and mechanical digestion that we have to do really can be rough. And think about some of the food that you eat might be very hot. It might be a little sharp even, depending on, if it's like a potato chip or something. And so it can scrape off those epithelial cells. So they're replaced every 10 days. They do have taste hairs, which protrude from openings called from the taste pores, and they are the sensitive portions of the cells. One of the benefits of saliva, of the watery portion of saliva, is that it dissolves those chemicals, so that way we can actually taste something. When your mouth is dry, you don't taste things as well as if your mouth is wet. If you're ever curious about that, do it with like a saltine cracker. That's a classic test. Sense of taste involves specific membrane protein receptors similar to the sense of smell, olfaction does, binding with the chemicals in food. And taste cells are rapidly adapting as well, so you can quickly shift what your perception of taste is. So here you can see those papilla in the tongue, mostly towards the root and the base of the tongue. They have all our taste buds. And something to be clear about is that, and I think this next slide might talk about it, your taste buds are scattered all over the dermal papilla, so it's not like you have one region for sweet, one for sour, one for umami. It's all a mixture completely. But you can see as you go along here, the sensory fibers with the receptors at the end. You have this epithelial tissue right here, part of the tongue, and then some connective tissue supporting it. Okay, so there are 5 types of taste cells in the taste buds. Again, each one is sensitive to a certain type of chemical stimulus, but they're mixed all around each other. So sweet, sour, salty, bitter, and umami, that savory flavor. Others, things like alkaline and metallic flavors. Taste buds responsive to one taste sensation with distinct receptors. All types of buds are found in all portions of the tongue, and taste sensation undergoes rapid adaptation like smell. Okay, hearing. So the ear provides a sense of hearing and equilibrium, so it does both of those things in this kind of shared structure. There are outer, middle, and inner portions of your ear. There is the external ear, which is the pinna, and that's the actual, the ear flaps when you think of your ear, right? It's those things. The external acoustic meatus, I always say the outside hearing hole. External acoustic meatus, your outside hearing hole. This is the canal. So if you're using a Q-tip like you're not supposed to and you put it in your ear canal, that's where that's the external acoustic meatus. That's what sound gets funneled towards until you reach the tympanic membrane, the eardrum. And this is the end of the acoustic meatus, and it vibrates with sound waves. So as sound is collected by the pinna, it gets funneled into the acoustic meatus, and then it stimulates and beats up against the tympanic membrane. Here you can see the auricle and tube and the tympanic membrane making that external ear, that outer ear. And then you get into some of these other structures, the middle ear with the incus, the malleus, and the stapes. That's what's transmitting sound waves from the tympanic membrane into the very inner ear, where you actually have the hearing structures for hearing and equilibrium balance. So the middle ear is that air-filled space. It has 3 tiny bones. These are the smallest bones, so they're called ossicles. The malleus, the incus, and the stapes. So the tympanic membrane vibrates against the malleus. It sits right up against it. That then sends its sensations to the incus, and then finally the stapes. And the stapes is pressed right up against the oval window of the inner ear, and it vibrates that fluid inside of it. That then stimulates the hearing receptors. We'll see that hearing receptors are basically hairs immersed in fluid, and so as that fluid vibrates, it gets transmitted as sound. These ossicles are going to transmit and amplify sound waves. So they aren't just there for sending the signal, they also help us to pick up on quieter sounds. They make them more obvious to us. There's, just for reference, the size of those little tiny bones. When we say small, we mean small. So the auditory tube, the eustachian tube, connects the middle ear to the nasopharynx, helps to maintain equal air pressure on both sides of the eardrum. So when your head gets stopped up or something, whether it's due to changes in pressure or maybe you have an ear infection or something, and you might pull on your ears and you might feel drainage down into your throat, that's the eustachian tube. Mucous membranes infections of the throat can travel up the auditory tube and cause middle ear infections. So that's why they pair the ear, nose, and throat doctors together, because an ENT can then be aware and treat that entire area. The inner ear, this is we call it the labyrinth, the bony labyrinth, and it's a communicating network of chambers and tubes. So a lot of this is based off of fluid being moved and shifted around in this inner ear labyrinth for us to be able to determine our position in space, where you are, how your head is being balanced, and what we're hearing. And I always think it's interesting because you're learning about hearing while you're listening to me, so it's just interesting to think about that. Hearing sound are just waves, vibrating particles through the air. Air transmits sound very well. Water does not transmit sound as well. That's why you can't, if you put your head underwater, it's all kind of gets really mumbly and muffled. And so it's because that sound wave is not being transmitted. And so when you're listening, when you're hearing me, what's happening is that inner ear, the membranous labyrinth inside of the bony labyrinth, inside your temporal bone, it has this fluid called perilymph, as well as endolymph inside the membrane. And the labyrinth contained the cochlea for hearing and the semicircular canals, which function in equilibrium. So here is that. So this is that cochlear and vestibular nerve. So that's going in towards the brain. So this is going to the deeper side. Out over here, this is going to be the more superficial area towards the middle ear. This oval window, this structure, is where the stapes synapse is with. So, okay, great, perfect, here you go. So we're talking right here in the inner ear. And so just so you can orient yourself, as you work this way, you're going towards the middle and the outer ear. So this portion over here, these are the semicircular canals, and so they have fluid that's running through them, that endolymph running through them, that will shift around and help you determine yourself in space. And then this is the cochlea, and cochlea to me just sounds like a round kind of word, cochlea. It has a lot of round sounds, round shapes, and so it's this little snail's tail right over here. You'll also hear this called the spiral organ, I believe is what some other people will call it. That's totally fine. This also is filled with fluid. It has what's called a basilar membrane, and so as the fluid, the endolymph shifts, it stimulates the basilar membrane in different ways. That shifts the hair, and then signals can be sent down the cochlear nerve that are interpreted as sound, the perception as sound. So here is seeing it within those semicircular canals, or I'm sorry, this is within the cochlea, my fault. So this is within the cochlea, it's just kind of taking it, cutting it in half. You can see this is the fluid, these are the hair cells, this is that membrane, and so they're being stimulated as fluid shifts for different sounds. Okay, so vibrations of the basilar membrane transmitted into the hearing receptor organ, the spiral organ that it rests on. The spiral organ runs the entire spiral length of the cochlea, so the cochlea is the bone portion, the spiral organ is the internal portion. Hair cells, those are the receptor cells of the hearing sense. Have those hairs that extend into the endolymph of the cochlear duct. The hair cells are on top of a tectorial membrane that touches the tips with the hair. As sound vibrations pass from the basilar membrane into the hair cells, it bends this membrane, and mechanical deformation leads to a generation of action potentials. So these are mechanical cells. These are mechanoreceptors. These are not chemoreceptors. You don't have to have a specific chemical binding to stimulate them. You don't have to have light like photoreceptors. These are going to be based off of mechanical deformation, so mechanoreceptors. Vibrations in that inner ear fluid cause different parts of cells to be stimulated and bent in different directions, turning them on or inhibiting their signals, and then that is what allows us to interpret it as sound. Action potentials get conducted along the cochlear branch of the vestibular cochlear nerve. So here you can see that again, that tectorial membrane on top of the basilar membrane with their supporting cells, the receptor cells, and these hair cells. Here it is, just an electron microscopic image. And this is that step by step, so that way you can, if you prefer to see it written out in each step, you can read through there. Okay, so then into equilibrium. Hearing and equilibrium are tied together, and that just is very helpful because let's say you hear a sound, off to the side. I'm just going to snap real quick. Off to the side, it can immediately, as you turn your head to listen for that sound, you can know where you are in balance, so you don't lose your position and you don't, and it does happen, right? As we get older, you turn quickly to look for a signal, and it might make you a little dizzy at first. I'm thinking roller coasters, right? If you go on a roller coaster and it's like just for a few minutes after, you're just still things are kind of still swirling around. If it hasn't happened to you yet, it probably will. I'm sorry. But equilibrium helps us to maintain our sense of awareness of where we are. Static and dynamic equilibrium. So static equilibrium, those organs help to maintain position of the head, posture, and balance when our head and body are still, versus dynamic is when we're moving or rotating. Static organs are located within the vestibule of the inner ear. It's that space between the cochlea and semicircular canals. It has two chambers, the utricle and the saccule. Each one of those contains an organ called the macula that has hair cells, a gelatinous material, and otoliths. These are little calcified gelatinous little stones, basically. So as you turn your head and as you move, that gelatinous material with these little stones in it start to shift. And just like with hearing, they bend the hair cells, so these are mechanoreceptors, and that generates action potentials, which then travel up the vestibular branch of the vestibular cochlear nerve. So here you can see, again, you have the semicircular canals over there, the cochlea over here, and then right in the middle is this vestibule. And then you can see the macula with the otoliths and this gelatinous material and these hair cells. And as these hair cells, as our head moves, these otoliths are going to shift back and forth, and then that then bends the hair cells, and then that signal gets sent. Yes, it's again on-off signals get sent down the nerve roots, the nerve branches, and then it lets us know where our perception of where we are in space, of our head positioning. Organs of dynamic equilibrium, those are inside the semicircular canals. They detect the motion of the head. Really, that's what we're worried about. With the rest of our body, your body will orient itself to your head. So as long as you pretty much know where your head is in space, the rest of your body will orient itself to that. So mostly we're looking for our head. Proprioceptors help us to know where the rest of our body is. The organs of dynamic equilibrium are the cristae ampullaris and the ampulla of each semicircular canal. These are hair cells, again, going to be mechanoreceptors into a gelatinous dome called the cupula. Rapid turning or movement causes the semicircular canals to move around while the endolymph stays still. This means that the cupula starts to bend, and the hairs and hairs of the hair cells embedded within it start to bend. That then generates those action potentials, and then it's interpreted into positioning through the vestibular cochlear nerve. And again, when you have proprioceptors in combination with these equilibrium receptors, we get to know where we are in space. So you can see the cupula with its hair cells, and then as they're stimulated, you get this signals being sent through the sensory neuron. That's just another example of it. So depending on whether you're not, you're tilting your head or rotating your head, you have these three kind of looping canals, so it will always kind of be in all three planes, basically, X, Y, and Z planes. Okay, so let's get into sight. Also, the eye is the organ that contains visual receptors, and they provide the sense of vision. So these are going to be photoreceptors, not mechanoreceptors, not chemoreceptors. They're stimulated by light, and the light then causes them to produce or break down, depending on the stimulation of light, causes them to produce or break down a neurotransmitter. Accessory organs of sight, things like your lacrimal apparatus that helps to keep things nice and moist, eyelids, extrinsic muscles to help you look around. These are all these things are either providing assistance through protection or movement. The eyelid protects the eye from foreign objects. There are 4 layers to it: the skin, muscle, connective tissue, and the conjunctiva. The muscles, the orbicularis oris and levator palpebrae. So levator palpebrae, the thing that lifts your eyelid, is the kind of translation for that. So these are the muscles that help you to open or close your eyes, and then this is what's going to help you in positioning to look your eyes to look around. The conjunctiva is a mucous membrane, lines the inner surface of the eye and folds back to cover the anterior surface of the eyeball. So this is why if you ever wear contacts, your contacts can't go into the back of your head, because you have this conjunctiva that helps to basically seal it forward. Here you can see the basic eye diagram. You have the inner portion of the eye back here. Then you start to have structures like your lens, the cornea, and then right in here is the conjunctiva. And so you can see that the conjunctiva, yeah, there we go. We can follow it that way, and then you can follow it this way. And so it terminates in these little, kind of like a cul-de-sac. And so you kind of can go in, but then it will not let you go actually back into the eye. It's going to keep it forward. Yeah, you have the cornea, which is covering the outer eye. I'm trying to see, it almost looks like this eyelid is closed. Right? Yeah, there's the eyelashes. So this eyelid looks like it's nice and closed. All right, so the lacrimal apparatus, you have a lacrimal gland that is producing tears to help lubricate and cleanse the eye. Tiny tubes release tears over the surface of the eye. There are 2 ducts called the canaliculi that drain the tears into the lacrimal sac through the nasolacrimal duct into the nasal cavity. These tears also have lysozymes to help provide antibacterial protection. And so this is that tear duct, basically. Your eyes should stay nice and moist. And sometimes when the tear ducts aren't draining properly, or when there's an excess of moisture, let's say you're crying, it can't drain through the nose. Some of it will, right? Because your nose starts to run and you start to just kind of get very moist in your face. But also because you have producing so much water, your tear ducts actually will then start to leak out through your face because they can't drain fast enough through this sac and duct. The extrinsic eye muscles attached to the sclera, we'll see that's the outer covering of the eye. These have 6 extrinsic muscles to help move it in all directions. So here is that nasolacrimal duct, that lacrimal gland right in there. I'm sorry, over here is a lacrimal gland. It washes tears over your eye, and they drain from the medial portion down the nasal cavity. Here you can see those extrinsic eye muscles. We'll look at the intrinsic ones later, but extrinsic means on the outside. Intrinsic are going to be the ones that help to change the shape of the eye, as well as focusing the pupil. And sorry, going back real quick, you can see this outer covering, the white part is the sclera, and then the optic nerve in the most posterior portion. So the eye itself has 3 distinct layers, an outer, middle, and inner. The outer is the fibrous layer that's going to help give it the shape. The middle is the vascular layer, which has all the blood vessels, and the inner eye rather is the nervous layer, which is the retina. Also, the spaces contain fluids to help maintain shape. We'll see that. So here it is looking at again from the side view. You can see this posterior chamber as well as an anterior chamber. I'm sorry, cavity, that's correct. It's a cavity. So the anterior cavity defined by the lens to the cornea, and the posterior cavity is this entire portion back here. The anterior cavity is divided both into an anterior and posterior chamber, defined by the iris. We'll get into this later. We'll get into this more in detail in lab as well. But again, looking at the 3 layers of the eye, you have this outer fibrous layer, which is the sclera and cornea, And then you have this middle vascular layer, which is going to have things like the choroid, the ciliary body and choroid body. This is where the blood vessels are, and then it also becomes the iris. That's the colored portion of the eye. It has the color because it's vascularized. It actually has blood vessels. And then the inner layer is this retinal layer, this yellow layer, and you can see it forms back in here into the optic nerve. So this is actually the photosensitive portion of it. And then each of these cavities, the posterior and anterior cavity, are, like we said, fluid filled. And so if somebody has things like glaucoma, they test the pressure of the eye. A lot of times it's because these fluids are getting to be too high pressure. So the outer fibrous layer, the cornea and sclera, continuous with each other. The cornea is the transparent anterior portion of the outer layer of the eye, helps to focus light rays, but it's completely transparent versus the white part of the eye, which is the sclera, and that is obviously not. The sclera is white, and it's the posterior 5/6 of the outer layer. Mostly collagen, protects the eye, and an attachment site. Optic nerves and blood vessels go through the sclera to the posterior eye. This middle vascular layer is the choroid coat, the ciliary body, and the iris. The choroid coat is vascular, darkly pigmented. It helps to nourish other tissues of the eye and to keep the inside of the eye dark. So it helps to lessen light pollution, basically, from and rather have it all focused in through the iris or the pupil. The ciliary body forms a ring around the front of the eye. It has the ciliary processes and the suspensory ligaments that hold the lens in position and change its shape as we focus at at images of different distances. So if you're focusing on something close or something further away, your lens has to change the shape, and so the ciliary bodies and those help to do that. Here you can see the ciliary processes of the ciliary bodies. Right, these are the ciliary bodies out here, the processes and the ligaments that help to hold the lens right in space and also change its shape. You then have the retina, the choroid, and the sclera. Still in the middle layer, the ability of the lens to adjust shape is called accommodation. So when the lens changes its shape to focus on something, it's accommodating to those things. The ciliary muscles relax. The ligaments pull outward, and the lens flattens for distant objects. So the flatter the lens, the easier it can see distant objects, versus taking on a more convex shape to view closer objects as the ciliary muscles contract. As you age, your eyes might not get as good at accommodating, and so you use lenses or contact lenses, glasses or contact lenses, to then help your eyes so they don't have to accommodate as much. A lot of times the reason why people say you should go to the eye doctor every year is because you may see fine, you may be able to see, but your eyes might be having to work really, really hard to help you see, to accommodate your eyes enough to be able to see. And so it can sometimes cause headaches and strain and tension, merely just trying to see, because these muscles are working constantly to try to keep your lenses accommodated enough to focus on what you're focusing on. And so glasses help to let those muscles relax. This is just showing that accommodation versus when the ciliary muscles are contracted versus relaxed. The iris then, this is a thin pigmented diaphragm of smooth muscle, the colored portion of the eye lying between the cornea and the lens. It adjusts the amount of light entering the pupil, an opening in the center of the iris. So this is the colored portion of the eye. It's going to, people might think that the pupil grows and contracts, but really it's your iris is contracting or dilating. Dilating or constricting. So as you go into brighter light, your iris is going to constrict and make the pupil smaller. Or if you go into very dim low lighting, your iris is going to dilate, and that allows the pupil to open up more. The aqueous humor is secreted by those ciliary bodies, filling the space between the cornea and the lens, nourishing cells and keeping its shape. So here you can see a sympathetic nerve fiber. Sympathetic stimulation is going to cause dilation, and so larger pupil, versus parasympathetic innervation, which causes constriction and a smaller pupil and a bright light. This is one of those things that may accommodate over time. So as you walk into a dimly lit room, your eyes will over adjust. So if you're going, especially if you're going from outside bright into a dark room, your eyes will really open up. And then as they get adjusted to it, they start to see, oh no, there is some dim light in here. They might go back to a slightly more constricted version, or probably somewhere in between, most likely. Then the inner layer, this is the actual photosensitive layer of the eye. This is the retina with its photoreceptors. Those are the visual receptor cells. It consists of several layers of cells continuous with the optic nerve in the back of the eye, and it forms the inner lining of the eye. The center of the retina is the macula lutea, and this also has the fovea centralis, and this is the point of sharpest vision in the retina. We'll look at the place where is it, right here, where it says the continuous with the optic nerve. That actually, right in the very back of the eye, that's the optic disc. That is where you have the, that's your blind spot, right where the opening of the optic nerve meets the retina. There are no photoreceptors there, and so that's the blind spot, versus the spot right off to the side of it, this fovea centralis, is a very, very sharp vision. So this is just showing how photoreceptors are arranged to receive and be stimulated by light. So this is the external portion. This is the more external. You can see light is coming in through the iris, through here, passing through the vitreous humor, and then it stimulates these different cells. So you have this sclera, which is a very outer portion of the eye, so because it's the back of the eye, this pigmented choroid, and then right along here you have these the photoreceptors themselves. So as light comes in, it's going to stimulate the nerve fibers, or the nerve cells. It's going to stimulate the photoreceptors and send a signal along. Passing through. Here we go. Medial to the so this is what I was talking about before. Medial to the fovea centralis is the optic disc. That's the blind spot because there's no photoreceptors in the area. That's where the optic nerve is meeting the retina. The posterior cavity has its vitreous humor versus the aqueous humor in the anterior cavity, giving its similar functions, supporting the shape, maintaining nutrients. Here you can see the optic disc, that blind spot, and then just medial to it, the macula lutea with the fovea centralis. And there it is, just on an actual picture. Okay, light refraction, corrective lenses. So focusing causes light rays to bend, which causes this refraction. Refraction causes images to fall on this fovea centralis, so it basically allows them to, our eye can focus the image specifically on the fovea centralis to make sure that we're seeing it the most clearly. That's why, like, when you look at something, maybe you have to like look at it again and kind of squint potentially. Your body kind of naturally will adjust. You might tilt your head to the side a little just to try and get to see the thing just right. And what's happening is that your body is trying to center it on the fovea centralis. The cornea and the lens refract light rays to help focus things. Different issues that might happen, emmetropia, sure. This is normal vision, myopia, light rays focused and scatter, causing a blurry image, and hyperopia, where light rays are focused to the back of the retina but not yet converged when they reach, and so you have a convex lens to correct that. That's just showing how light rays are adjusted. Okay, let's get into this actual transmission of these signals. So the photoreceptors are both rods and cones. They're the two types of modified neurons, which are your visual cell receptors. In our bodies, we have one type of rod and three types of cones. Species like birds, I think, have sometimes up to 27 or 80 different types of cones. So they have a very different type of vision than we do. Rods are more sensitive to black and white and really are just there to help detect light. So they are there just to try to give some type of illumination to our sense of being and into our world. Less precise images, mostly just general outlines, and more rods than cones. Cones provide sharp images and bright light, so things need to be well lit. Otherwise, our brain just kind of fills in the images. Because, especially if you're in a place that you're familiar with, you might see a lump and be like, oh yeah, I know what that is. That's that thing. And automatically you might start to, add a little more sharpness to that image. Cones also provide color vision, and these are mostly located in the fovea centralis. Densely packed cones, no rods in that portion of the eye, though. Okay, this is just, again, showing that orientation of them. There's an electron image. So rods and I'm going to do that every time. Rods and cones have light-sensitive pigments breaking down upon the absorption of light energy. So this is rhodopsin. It's the light-sensitive pigment in rods. When rhodopsin gets stimulated by light, it breaks apart into opsin and retinal. So that comes from our vitamin A. Decomposition activates an enzyme that initiates changes in the rod cell membrane that generates a nerve impulse. So as rhodopsin is broken down, it basically then allows this enzyme to become active, because it was inhibiting that enzyme before. But once the rhodopsin is broken down, the enzyme becomes active, and then it allows a nerve impulse to be generated. Those impulses leave the retina through the optic nerve, travel to the visual cortex and the occipital lobe, and get interpreted and processed as vision. That also, is going to activate different parts of the brain to help provide, things like memory and higher reasoning, higher thinking, or cognitive perception of what something we think something should be will also help to change what it is. In bright light, almost all the rhodopsin gets broken down. In dim light, regeneration is faster than breakdown, and so things can be harder to see. Those axons of the retinal neurons leave the eyes and form the optic nerves. Those then cross over that point of convergence at the optic chiasma, and they go from there into the thalamus. Just before reaching the thalamus, some are going to go into the function and visual reflexes. So if there's something that we need to that is identified immediately as a dangerous thing, or something that needs to be refocused on, right? So if it's like there's something that's moving really quickly, sometimes you might just follow it on instinct, right? It feels like it's on instinct. It's a reflex. From the thalamus, they then go to the optic radiations of the visual cortex. Here you can just follow those pathways. Again, left is going towards the right, right going towards the left. There's where they cross over at the chiasma. Right. Thank you. Hope you enjoyed that lecture.